The I-squared sheet is the second form of regular secondary structure in proteins. Beta sheets consist of beta strands connected laterally by at least 2 or 3 backbone hydrogen bonds, forming a generally twisted, pleated sheet. A beta strand is a stretch of polypeptide chain, typically 3 to 10 amino acids long, with backbone in an extended conformation. The higher level association of I-squared sheets has been implicated in formation of the protein aggregates and fibrils observed in many human diseases, notably the amyloidosis such as Alzheimer's disease. History: the first I-squared sheet structure was proposed by William Asbury in the 1930s. He proposed the idea of hydrogen bonding between the peptide bonds of parallel or antiparallel extended I-squared strands. However, Asbury did not have the necessary data on the bond geometry of the amino acids in order to build accurate models, especially since he did not then know that the peptide bond was planar. A refined version was proposed by Linus Pauling and Robert Quarry in 1951. Structure and orientation. Geometry. The majority of I-squared strands are arranged adjacent to other strands and form an extensive hydrogen bond network with their neighbors, in which the NH groups in the backbone of one strand establish hydrogen bonds with the C=O groups in the backbone of the adjacent strands. In the fully extended I-squared strand, successive side chains point straight up, then straight down, then straight up, etc. Adjacent I-squared strands in a I-squared sheet are aligned so that their psi plus or minus atoms are adjacent and their side chains point in the same direction. The pleated appearance of I-squared strands arises from tetrahedral chemical bonding at the psi plus or minus atom. For example, if a side chain points straight up, then the bond to them must point slightly downwards, since its bond angle is approximately 109.5 degree. The pleating causes the distance between and to be approximately 6 rather than the 7.6 expected from two fully extended trans peptides. The sideways distance between adjacent psi plus or minus atoms in hydrogen-bonded I-squared strands is roughly 5a. However, I-squared strands are rarely perfectly extended. Rather, they exhibit a twist due to the chirality of their component amino acids. The energetically preferred dihedral angles near equals diverge significantly from the fully extended conformation equals. The twist is often associated with alternating fluctuations in the dihedral angles to prevent the individual I-squared strands in a larger sheet from splaying apart. A good example of a strongly twisted I-squared hairpin can be seen in the protein BPTI. The side chains point outwards from the folds of the pleats, roughly perpendicularly to the plane of the sheet. Successive residues point outwards on alternating faces of the sheet. Hydrogen bonding patterns because peptide chains have a directionality conferred by their N-terminus and C-terminus, I-squared strands too can be said to be directional. They are usually represented in protein topology diagrams by an arrow pointing toward the C-terminus. Adjacent I-squared strands can form hydrogen bonds in antiparallel, parallel, or mixed arrangements. In an antiparallel arrangement, the successive I-squared strands alternate directions so that the N-terminus of one strand is adjacent to the C-terminus of the next. This is the arrangement that produces the strongest interstrand stability because it allows the interstrand hydrogen bonds between carbonyls and amines to be planar, which is their preferred orientation. The peptide backbone dihedral angles are about in antiparallel sheets. In this case, if two atoms are adjacent in two hydrogen-bonded I-squared strands, then they form two mutual backbone hydrogen bonds to each other's flanking peptide groups. This is known as a close pair of hydrogen bonds. In a parallel arrangement, all of the N-termini of successive strands are oriented in the same direction. This orientation may be slightly less stable because it introduces non-planarity in the interstrand hydrogen bonding pattern. The dihedral angles are about in parallel sheets. It is rare to find less than 5 interacting parallel strands in a motif, suggesting that a smaller number of strands may be unstable. However, it is also fundamentally more difficult for parallel I-squared sheets to form because strands with N and C termini aligned necessarily must be very distant in sequence. There is also evidence that parallel I-squared sheet may be more stable since small amyloidogenic sequences appear to generally aggregate into I-squared sheet fibrils composed of primarily parallel I-squared sheet strands, where one would expect anti-parallel fibrils if anti-parallel were more stable. In parallel I-squared sheet structure, if two atoms are adjacent in two hydrogen-bonded I-squared strands, then they do not hydrogen bond to each other. Rather, one residue forms hydrogen bonds to the residues that flank the other. For example, residue may form hydrogen bonds to residues and this is known as a wide pair of hydrogen bonds. By contrast, residue may hydrogen bond to different residues altogether, or to none at all. Finally, an individual strand may exhibit a mixed bonding pattern, with a parallel strand on one side and an antiparallel strand on the other. Such arrangements are less common than a random distribution of orientations would suggest, suggesting that this pattern is less stable than the anti-parallel arrangement. However, bioinformatic analysis always struggles with extracting structural thermodynamics since there are always numerous other structural features present in whole proteins. Also, proteins are inherently constrained by folding kinetics as well as folding thermodynamics, so one must always be careful in concluding stability from bioinformatic analysis. The hydrogen bonding of I-squared strands need not be perfect, but can exhibit localized disruptions known as beta bulges. The hydrogen bonds lie roughly in the plane of the sheet, with the peptide carbonyl groups pointing in alternating directions with successive residues. For comparison, successive carbonyls point in the same direction in the alpha helix. Amino acid propensities equals, large aromatic residues and I-squared branched amino acids are favored to be found in I-squared strands in the middle of I-squared sheets. Interestingly, different types of residues are likely to be found in the edge strands in I-squared sheets, presumably to avoid the edge-to-edge association between proteins that might lead to aggregation and amyloid formation. Common structural motifs. A very simple structural motif involving I-squared sheets is the I-squared hairpin, in which two antiparallel strands are linked by a short loop of 2 to 5 residues, of which one is frequently a glycine or a proline, both of which can assume the dihedral angle conformations required for a tight turn or a beta bulge loop. Individual strands can also be linked in more elaborate ways with longer loops that may contain alpha helices. Greek key motif equals. The Greek key motif consists of 4 adjacent antiparallel strands and their linking loops. It consists of 3 antiparallel strands connected by hairpins, while the fourth is adjacent to the first and linked to the third by a longer loop. This type of structure forms easily during the protein folding process. It was named after a pattern common to Greek ornamental artwork. The I-squared I plus or minus I-squared motif equals. Due to the chirality of their component amino acids, all strands exhibit a right-handed twist evident in most higher-order I-squared sheet structures. In particular, the linking loop between two parallel strands almost always has a right-handed crossover chirality, which is strongly favored by the inherent twist of the sheet. This linking loop frequently contains a helical region, in which case it is called a I-squared I plus or minus I-squared motif. A closely related motif called I-squared I plus or minus I-squared I plus or minus motif forms the basic component of the most commonly observed protein tertiary structure, the TIM barrel. I-squared meander motif equals, a simple supersecondary protein topology composed of 2 or more consecutive antiparallel I-squared strands linked together by hairpin loops. This motif is common in I-squared sheets and can be found in several structural architectures including I-squared barrels and I-squared propellers. Psi-loop motif equals, the psi-loop, I-loop, motif consists of 2 antiparallel strands with one strand in between that is connected to both by hydrogen bonds. There are 4 possible strand topologies for single I-loops as cited by Hutchinson et al. This motif is rare as the process resulting in its formation seems unlikely to occur during protein folding. The I-loop was first identified in the aspartic protease family. Structural architectures of proteins with beta sheets, Beta sheets are present in all I², I plus or minus plus I² and I plus or minus /I² domains and in many peptides or small proteins with poorly defined overall architecture. All I² domains may form I² barrels, I² sandwiches, I² prisms, I² propellers, and I² helices. Structural topology. The topology of a I² sheet describes the order of hydrogen-bonded I² strands along the backbone. For example, the flavodoxin fold has a 5-stranded, parallel I² sheet with topology 21345. Thus, the edge strands are I² strand 2 and I² strand 5 along the backbone. Spelled out explicitly, I² strand 2 is H-bonded to I² strand 1, which is H-bonded to I² strand 3, which is H-bonded to I² strand 4, which is H-bonded to I² strand 5, the other edge strand. In the same system, the Greek key motif described above has a 4123 topology. The secondary structure of a I² sheet can be described roughly by giving the number of strands, their topology, and whether their hydrogen bonds are parallel or antiparallel. I² sheets can be open, meaning that they have 2 edge strands, or they can be closed beta barrels. I² barrels are often described by their stagger or shear. Some open I² sheets are very curved and fold over on themselves or form horseshoe shapes. Open I² sheets can assemble face-to-face or edge-to-edge, forming one big I² sheet. Dynamic features. Beta sheets in proteins may carry out low-frequency accordion-like motion as observed by the Raman spectroscopy and analyzed with a quasi-continuum model. Parallel I² helices. A I² helix is formed from repeating structural units consisting of 2 or 3 short I² strands linked by short loops. These units stack atop one another in a helical fashion so that successive repetitions of the same strand hydrogen bond with each other in a parallel orientation. See the beta helix article for further information. In left-handed I² helices, the strands themselves are quite straight and untwisted. The resulting helical surfaces are nearly flat, forming a regular triangular prism shape, as shown for the 1-QRER carbonic anhydrase at right. Other examples are the lipidase synthesis enzyme LPXA and insect antifreeze proteins with a regular array of THR-SIDA chains on one face that mimic the structure of ice. Right-handed I² helices, typified by the pectate lyase enzyme shown at left or P22-phage tail spike protein, have a less regular cross-section, longer and indented on one of the sides. Of the 3 linker loops, one is consistently just 2 residues long and the others are variable, often elaborated to form a binding or active site. A 2-sided I² helix is found in some bacterial metalloproteases. Its 2 loops are each 6 residues long and bind stabilizing calcium ions to maintain the integrity of the structure, using the backbone and the asp side chain oxygens of a GGX-GXD sequence motif. This fold is called a beta roll in the SCOP classification. I² sheets in pathology. Some proteins that are disordered or helical as monomers, such as amyloid I² can form I² sheet-rich oligomeric structures associated with pathological states. The amyloid I² protein's oligomeric form is implicated as a cause of Alzheimer's. Its structure has yet to be determined in full, but recent data suggest that it may resemble an unusual 2-strand I² helix. The side chains from the amino acid residues found in a I² sheet structure may also be arranged such that many of the adjacent SIDA chains on one side of the sheet are hydrophobic, while many of those adjacent to each other on the alternate side of the sheet are polar or charged, which can be useful if the sheet is to form a boundary between polar watery and nonpolar greasy environments. See also, folding, tertiary structure, I plus or minus helix, collagen helix, foldamers. References. Further reading, Cooper, J. Supersecondary Structure, Part 2, May 31, 1996. Accessed May 25, 2007. Structural Classification of Proteins Open-Sided Beta Meander, October 20, 2006. Accessed May 31, 2007. External links, Anatomy and Taxonomy of Protein Structures Survey. NetSurfP, secondary structure and surface accessibility predictor, interactive model of an anti-parallel I-squared sheet, interactive model of a parallel I-squared sheet, animated details of I-squared pleated sheet, Munwuxignes.